Well, hello, everyone. Thank you so much for joining our webinar today. We're very excited at Science Interactive to be launching our new genetics discipline. We're going to give everyone just a few minutes to get logged in today. And while we're doing that, we're going to put up a poll question so we can better understand who our audience is. So our first question is, how do you currently deliver your genetics lab? So please take the time to answer this poll question. We'll go over the results in just a moment. All right, well, looks like the results are coming in now, and about 50% of our audience today teaches face-to-face labs on campus. A little over a third use lab kits. And then the remainder of our audience, and this is great, they're not currently teaching a genetics lab. So, maybe today we can inspire you for how that's possible, and you might join the crew, so to speak. All right, we're going to give everyone about one more minute to get connected, and then we'll get started. As you're getting connected, just some housekeeping rules for our very short 30-minute session today. We do encourage you to ask questions, and you will be muted. However, you can use the chat tool or the Q&A tool on your Zoom bar to pose those questions. Now, because I'm the only presenter today, I apologize. While I may see your question come in with a little pop-up notification on my screen, I will not be able to read your question as I'm presenting the material today. However, I will ensure you that we will end with plenty of time to address those questions that have come over during the presentation. So again, if you have a question, please feel free to ask. Again, thank you for joining, everyone. We appreciate you taking time out of your schedule today to learn more about the Science Interactive launch of genetics labs. We're very excited about this. We know you're busy this time of year, whether you're just getting back in to work to begin on the fall semester, or maybe you're trying to tie up some loose ends, such as grading and submitting those grades for your summer courses. So thank you very much. We do appreciate your time and interest. As promised today, we want to go over how to deliver engaging, high-quality genetics labs. And first of all, let me introduce myself. My name is Dwayne Cagle. I am the senior scientist at Science Interactive. And for the past 12 and a half years, I've been very fortunate to be able to facilitate the efforts of online science educators and their students. And today, like I said, I'm very, very excited to share with you the launch of our genetics laboratory curriculum and labs themselves. Now, when we started developing this project in January of this year, we were very mindful at that time of the results of our 2023 annual lab report on online science. And what we did last year is we surveyed over 1,600 online science students and their instructors to better understand their feelings about online laboratory instruction in general. And the students overwhelmingly responded that online laboratories were very important for them to be able to take science courses in general and to complete their degrees. We also wanted to get an idea of what kind of challenges instructors faced when teaching science courses online. And what we learned is 41% of instructors stated that recreating a hands-on lab experience was the #1 challenge they faced when teaching an online lab. About a quarter of instructors stated that the quality and rigor of the course materials were a challenge when teaching online labs. And one of the primary reasons that instructors were hesitant for even considering an online laboratory offering had to do with cost, not surprisingly. Well, let's look at the reality. The reality, though, is that 71% of instructors that used hands-on laboratory experiments and materials realized that they were comparable to the on-campus face-to-face experience. And indeed, 74% of students agreed. 89% of those instructors that used hands-on laboratory materials stated that students had sufficient opportunity to learn the knowledge and skills required within their given program. 84% of students agreed. And lastly, 72% of students stated that the cost of their online laboratory materials was aligned with the value of those items. Now, we were also interested in this survey of determining the value and importance of virtual simulations as opposed to only physical hands-on materials. And we posed that question in the survey. Both 73% of students and instructors agreed that virtual labs, when performed properly, could give students the opportunity to learn the knowledge and skills required in their programs. Furthermore, 91% of students and 74% of instructors agreed that having the opportunity to perform these virtual labs made the course more engaging for students. So what is it about online labs in general that students find engaging? And what do they feel should be the focus when developing labs to improve how engaging they are? And students highlighted the benefits as related to 4 primary categories. One, being able to participate in an immersive learning experience versus an on-campus lab where sometimes one or more students would feel excluded because they were always working in groups. And as we always know, every group has at least one student that really takes initiative, and sometimes the others, don't feel like they are participating equally. Number 2 is being able to engage more deeply and interact with the materials in a more sufficient manner. Again, if a student is in an online environment, they have more time and they can work at their own pace, as we see in point 3, likewise, versus again, being on campus where maybe someone's taking the lead and they're not that individual and they don't feel like they have equal access or sufficient time. And then lastly, and very importantly, giving students an opportunity to repeat procedures and experiments as needed in a low stakes environment. Again, meaning if they don't get sufficient results the first time around, not all will be lost, as sometimes they may feel like in a face-to-face lab. So when we look at engagement and improving that, that overcomes one of the major challenges that students have. But we'd like to ask a second poll question right now to instructors and to find out what you feel your biggest challenge is to offering genetics labs in general. Okay, thank you for the responses. So it looks like overwhelmingly student preparedness is the biggest concern and challenge that you have in the genetics courses that you teach. In student engagement being a close second. And that's very, very interesting. And I want to show you something that we're going to hopefully address that preparedness need through some of the products that we offer. But thank you again for your input and responses. So That leads us to how do we deliver high quality genetics labs to meet the demand that you, the instructors, have just expressed regarding student preparedness, and the demands that students expressed in our 2023 survey, in which engagement what was one of their biggest concerns and demands. So we addressed three overall factors and concerns when developing our genetics content. One, we wanted to make sure that we deliver hands-on labs in an online modality. It's so that all students have equal access to the curriculum. And we'll see in an upcoming demo preview that we're all going to get to experience here in just a couple of moments, how we make that happen through our SI Cloud platform. Also, we want to be able to support hybrid learning. We know not all programs are fully online. So we want to make both our lab manual and our materials flexible by nature and able to be used by both on-campus students or remote students, and have those fit into your program any way possible. And then, very importantly, and this is going to address the instructor concerns from our vote just 2 minutes ago, we want to be able to enrich on-campus labs. And to do that, we offer virtual pre-labs that better prepare students for the hands-on activities they will be performing once they're on campus, so they can make the most of that limited amount of time that they have available for that face-to-face laboratory experience. Now, when we design our curriculum, it is very important for us to follow best practices for online digital instruction. Many of you in our audience today are familiar with Quality Matters. And we have submitted not only our new genetics curriculum, but all of our disciplines to Quality Matters. And we have achieved certification across the board for all 14 of those disciplines. And as you can see, our library consists of over 450 total experiments. So whether you're teaching strictly genetics or you're looking to integrate some genetics labs into your overall biology course, or even your anatomy physiology offerings, you can be assured that all of these courses are QM certified and will require no additional effort on you, the instructor's end, to achieve certification on behalf of your institution. So what is the overall goal of the genetics labs that we have designed? Well, first and foremost, again, we want them to be able to enrich on-campus, hybrid, and online courses equally. We want to prep online students with digital pre-labs. So again, they are prepared when they get to that physical lab and they're ready to conduct their supervised experiments. We want to build practical skills for careers in health sciences. We know many of the students that use our kits and our curriculum are on a career pathway for nursing or other affiliated health science fields. And we know it's very important that those students have exposure to numerous topics within their field, but also they get to experience a variety of protocols for using various tools of their trade. And we'll see examples of that again in our genetics demo. And then lastly, we want to supplement digital labs with hands-on lessons and experiences for students. And that certainly comes into play in our library of over 450 experiments. Now, specifically for our genetics, offering, we elected to go fully digital in the 8 new labs that are now placed in our library. And there are a few reasons for that. One, we wanted to make them very easy to implement into existing courses for instructors. So you can easily plug these in to your course via your LMS. You don't need to worry about your students having to acquire any types of materials. So very easy to get started with this. Because they're digital, the pricing is very affordable for students because they're not having to purchase high-tech lab equipment. Certainly, if we're talking about the molecular aspects of genetics, that would traditionally only get to be experienced by on-campus students. And then again, we're including 8 virtual labs with the accompanying curriculum that has been certified by Quality Matters. And then lastly, and certainly not leastly, we wanted to ensure that our lab course delivery platform can be integrated into your current LMS. And this gives students step-by-step guidance and support. We also provide support for the integration itself. And very, very importantly, due to the integration, it saves you time by allowing our platform to auto-grade student responses and then automatically port them into your grade book within your learning management system. So overall benefits for instructors for our new genetics offerings is flexibility, course enrichment, student preparation, preparedness, ease of implementation, affordability, and then minimal safety concerns. Benefits for students, again, it's very affordable. Students don't have to keep track of complex equipment. Students can repeat procedures as many times as needed, so they feel comfortable with the activities that they're performing and gain a better understanding. Students get hands-on practice to learn how to use certain pieces of lab equipment. So again, once they get to an environment, whether it's an on-campus environment or a future environment, which they will require less training than students who haven't had an opportunity to practice these techniques in a digital modality. And then lastly, as we just mentioned, the ability to practice, very important for students. So here are the 8 titles that we're currently launching, and you can see that they range from molecular genetics to patterns of inheritance and karyotyping, all the way to population genetics and phylogenetics. So as promised, what we want to do now is give you a preview demo of an actual lab that we've just launched. That is the DNA extraction, amplification, and analysis lab. So if you'll bear with my computer just a moment, we do need to change the screen that we're viewing. And we should be able to see it now. Here we see all 8 of those labs listed. This is on our course delivery platform that I've already referenced, the Science Interactive Cloud. We're going to select the, again, DNA extraction, amplification, and analysis lab. We'll see it's broken down into 3 sections titled Exploration, the exercises, and then the evaluation section. These sections are gated for your students, but as instructors, we can jump around as we see fit. So you're getting the behind the scenes instructor tour of Cloud. So here we go. So let's take a look at the exploration section. And the exploration section is going to provide the student with the traditional background content within their lab manual. So this is going to review all the important concepts the student should understand before proceeding with the procedures. It has, in this lab, 3 learning objectives: to describe the molecular structure of DNA and how it's extracted from cells, to discuss the polymerase chain reaction and the in vitro amplification of DNA, and explain gel electrophoresis and how DNA sequencers are used to analyze DNA. Now, for time's sake, we're not going to go through this whole lab step by step. I will share with you that students begin the exploration section by taking a test your knowledge formative assessment. It helps them to identify any knowledge gaps that they have before they engage with the content. Now, the content itself is arranged by pages, each with a unique title and a unique set of concepts addressed. So if we select the PCR page here, we'll see there's a manageable block of text, followed by an interactive image. And these are very engaging for students. Here we see, if we select each step of the polymerase chain reaction, a new image appears along with the text panel. And therefore, the content is segregated by step in very manageable chunks of information that is easy for the student to comprehend. And I really need to give a shout out to the design team at Science Interactive. They're integral in developing these types of elements in all of our labs. And it really does show our focus to improving student engagement. Once a student completes the exploration section, and by the way, they do answer a series of questions at the bottom of each page to test their knowledge gain as they're completing each page of content, then the student is ready to move to the experimentation. And this is where they perform the actual exercises. There are unique learning objectives associated with this section, such as extract DNA from a mouth swab using a virtual lab bench, to amplify extracted DNA sequences using PCR simulation, and lastly, to analyze amplified DNA using gel electrophoresis and DNA sequencer animations. So let's take a look at one of these exercises. And you'll see here that a student is prompted to open a digital molecular lab bench and then proceed by very detailed step-by-step instructions that also visually guide the student through the virtual lab bench procedures. Now, this lab bench is very interactive. It contains over 200 DNA samples from different species. You can see the list here. It also contains an exhaustive list of primers that students will use for the amplification of those DNA sample sequences. The laptop itself contains a pipetter, a tube rack. Students are able to label each of these tubes as they place them from the sterile tube jar. Here is a thermocycler machine where students perform the PCR. There's a gel box that students will use to perform gel electrophoresis, and a DNA sequencer here, where students can compare sequences between 2 or more species. Now, we're not going to, again, for time's sake, go physically through an exercise, but I want to give you a visual of the tools that students are using to complete this particular procedure. Once a student completes their work, they're first asked to provide proof of completion. And to do that, in this particular instance, the student is going to take a screenshot of their completed gel, and then they're going to drag and drop that image into this photo panel. They're also going to measure the base pair lengths of each of the separated DNA sequences on that gel, and then we're going to record those base pair measurements within this automated data table. And then lastly, students are going to answer a couple of open-ended short answer questions. The last section of this in all of the genetics labs is titled evaluation. And in the evaluation section, students are going to review the previous learning objectives, and then they're going to answer a series of auto-graded multiple choice questions that span the breadth of all the information that was presented in this lab, from the exploration section to the procedures to the results of those procedures. And then lastly, students are going to be given an opportunity to answer an extension question. And this gives the students a chance to apply their knowledge to a unique scenario and problem. And these types of questions are very, very important, not only for students to demonstrate their true understanding of the concepts, but also for us to assess what a student has really learned, and can they apply that new knowledge gain. So let's go back. So that concludes our demo today. And as promised, we want to take the final couple minutes to address any of the questions that you've asked, and also to pose one last question for you. We would love to get in touch with you to provide you with detailed demos, access to some sample labs, and really work with you to tailor a lab solution that best meets the needs of your particular course. So please feel free to answer appropriately to this last poll question, and I'll review some of the questions that have come over. So I have a question to ask about accessibility in general, and whether or not we can mix digital labs with hands-on experiments, and vice versa. First of all, we always want to ensure that all of our curriculum and content is ADA compliant and therefore accessible to all students. So we have the requisite tools built into the back end of our curriculum delivery platform, and the types of images and interactives that we design and house within our curriculum are all ADA compliant. Also, yes, you can mix and match any number of fully digital or hands-on experiments within the curriculum, and therefore kit for your lab students. We do not have any kind of hard and fast rules against mixing and matching when it comes to those types of things. I have a question about how are these any different from McGraw-Hill online labs? Well, these are quite different in how they're designed. McGraw-Hill, what I have found, and you're welcome to correct me because I apologize, I haven't looked at them in about 24 months. But the last time I looked, they were very much of the nature of everything has to be selected in only one sequence to get the answers from the virtual lab bench experience that they designed. The lab bench that I gave you a preview of today, you notice we had very detailed instructions, and we visually guided a student through that. And that's because on that particular lab bench, a student can really do anything they want. They could choose any primer, whether it was the right one or not. They could choose reagents that were correct or not. They could select the wrong DNA sample to begin with, and it would greatly compromise the results the students received. So in essence, students have the ability to mess up, and it does affect their end result, which is pretty different than most of the virtual lab benches I've previewed in the past. And I apologize if McGraw-Hill's has changed greatly in the past 24 months, then I stand corrected. I just see one comment from a guest today regarding hands-on materials in general, that they have had challenges on their end. This is probably to an earlier poll question about quality hands-on materials for remote labs, and that is certainly a fair concern. Certainly, that was another guiding force in us opting to go fully digital on these genetic offerings, simply because I can't find, for example, an affordable micropipetter, an affordable gel box that works and gives the correct kind of results. Goes without saying there aren't affordable from a student perspective thermocyclers or DNA sequencers. But nonetheless, those are always going to be concerns. Certainly, there are experiments outside of molecular genetics that we can provide high quality hands-on materials for, and we have done that in all of our other discipline offerings that you will find in our library. So rest assured, we can offer that to you with the exception of molecular genetics. Well, I think we're about out of time today. Again, thank you so much for joining us. And we will be in touch for all of you that requested feedback or follow-up. So thank you again today for your participation. And we look forward to facilitating your online teaching needs in the weeks and months to come. Take care.